What are DNA, RNA, and genetic code? DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, are both nucleic acids, which are essential macromolecules that carry genetic information. They play crucial roles in the storage, transmission, and expression of genetic information in living organisms. 1. DNA, deoxyribonucleic acid, structure. DNA is a double-stranded molecule that has a helical structure, resembling a twisted ladder or a double helix. There are nucleotides in each strand. Each nucleotide has a sugar molecule, deoxyribose, a phosphate group, and one of 4 nitrogenous bases. These are adenine, A, thymine, T, cytosine, C, and guanine, G. Function. DNA carries the genetic instructions for the development, functioning, growth, and reproduction of all living organisms. It is located in the cell nucleus in eukaryotic cells and in the nucleoid region in prokaryotic cells. 2. RNA, ribonucleic acid, structure. RNA is typically a single-stranded molecule, although it can form secondary structures. Like DNA, RNA is composed of nucleotides, but it has ribose sugar instead of deoxyribose and the nitrogenous base uracil, U, instead of thymine, T. Function. RNA plays a crucial role in protein synthesis. There are different types of RNA, including messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. mRNA carries the genetic code from DNA to the ribosomes, where proteins are synthesized. tRNA helps in the actual assembly of amino acids into proteins, and rRNA is a structural component of ribosomes. 3. Genetic code. The genetic code is a set of rules that specifies the correspondence between nucleotide triplets, codons, in mRNA and amino acids in proteins. Each codon consists of 3 consecutive nucleotides in the mRNA sequence, and each codon codes for a specific amino acid or serves as a start or stop signal in protein synthesis. The genetic code is universal, meaning that the same codons code for the same amino acids in nearly all living organisms. This universality is a fundamental aspect of the shared genetic heritage of all life on Earth. In summary, DNA carries the genetic information in a cell, RNA plays a crucial role in protein synthesis, and the genetic code dictates the rules by which the information in DNA is translated into the amino acid sequence of proteins. These 3 components are central to the functioning and inheritance of genetic information in living organisms. Thank you for watching. Summary. DNA and RNA are essential macromolecules that carry genetic information in living organisms. DNA is a double-stranded molecule with a helical structure, carrying genetic instructions for development, growth, and reproduction. RNA, on the other hand, is a single-stranded molecule with ribose sugar and uracil base. It plays a crucial role in protein synthesis, with different types including messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. The genetic code, a set of rules that specifies the correspondence between nucleotide triplets in mRNA and amino acids in proteins, is universal and a fundamental aspect of the shared genetic heritage of all living organisms. Here are some keywords and their meanings based on this content. 1. DNA, deoxyribonucleic acid, meaning the genetic material found in cells, consisting of a double helix structure made up of nucleotide sequences that encode genetic information. 2. RNA, ribonucleic acid, meaning a single-stranded nucleic acid involved in various cellular processes, including protein synthesis, with different types such as mRNA, tRNA, and rRNA. 3. Nucleotide, meaning the basic building block of nucleic acids is composed of a sugar molecule, a phosphate group, and a nitrogenous base. 4. Double helix, meaning the twisted ladder-like structure formed by 2 strands of DNA, held together by hydrogen bonds between complementary nucleotide pairs. 5. Adenine, A, meaning one of the 4 nitrogenous bases in DNA and RNA, forming a base pair with thymine, in DNA, or uracil, in RNA. 6. Thymine, T, meaning a nitrogenous base in DNA that pairs with adenine, forming a complementary base pair. 7. Cytosine, C, meaning a nitrogenous base in DNA and RNA that pairs with guanine, forming a complementary base pair. 8. Guanine, G, meaning a nitrogenous base in DNA and RNA that pairs with cytosine, forming a complementary base pair. 9. Ribose, meaning a sugar molecule found in the backbone of RNA nucleotides. 10. Deoxyribose, meaning a modified sugar molecule found in the backbone of DNA nucleotides. 11. Helical structure, meaning a three-dimensional twisted structure, as seen in the double helix of DNA. 12. Genetic instructions, meaning the information encoded in DNA that directs the development, functioning, and characteristics of an organism. 13. Nucleus, meaning a membrane-bound organelle in eukaryotic cells containing the cell's genetic material, DNA. 14. Protein synthesis, meaning the process by which cells build proteins based on the information encoded in DNA, involving transcription and translation. 15. Messenger RNA, mRNA, meaning RNA molecule that carries genetic information from DNA to the ribosomes for protein synthesis. 16. Transfer RNA, tRNA, meaning RNA molecule that transports specific amino acids to the ribosome during protein synthesis. 17. Ribosomal RNA, rRNA, meaning RNA molecule that is a structural component of ribosomes, where protein synthesis occurs. 18. Codon, meaning a sequence of 3 nucleotides in mRNA that codes for a specific amino acid during protein synthesis. 19. Amino acid, meaning the building blocks of proteins are linked together in a specific sequence to form a polypeptide chain. 20. Universal genetic code, meaning the set of rules specifying the correspondence between nucleotide triplets, codons, and amino acids in nearly all living organisms. Here are some questions for you based on this content. 1. Question: what is DNA? Suggested answer: DNA, or deoxyribonucleic acid, is the genetic material found in cells, containing instructions for the development and functioning of living organisms. 2. Question: describe the structure of DNA. Suggested answer: DNA has a double helix structure composed of nucleotides, each consisting of a sugar molecule, a phosphate group, and one of 4 nitrogenous bases: adenine, thymine, cytosine, or guanine. 3. Question: what is RNA, and how does it differ from DNA? Suggested answer: RNA, or ribonucleic acid, is a single-stranded nucleic acid involved in various cellular processes, including protein synthesis. Unlike DNA, RNA contains ribose sugar and uses uracil instead of thymine. 4. Question: what are nucleotides? Suggested answer: nucleotides are the basic building blocks of nucleic acids, consisting of a sugar molecule, a phosphate group, and a nitrogenous base. 5. Question: explain the concept of a double helix. Suggested answer: a double helix is the twisted ladder-like structure formed by 2 strands of DNA, held together by hydrogen bonds between complementary nucleotide pairs. 6. Question: name the 4 nitrogenous bases in DNA. Suggested answer: adenine, A, thymine, T, cytosine, C, and guanine, G. 7. Question: what is the role of adenine in DNA? Suggested answer: adenine forms a base pair with thymine in DNA, stabilizing the double helix structure. 8. Question: define protein synthesis. Suggested answer: protein synthesis is the cellular process in which proteins are built based on the information encoded in DNA, involving transcription and translation. 9. Question: where is DNA located in eukaryotic cells? Suggested answer: DNA is located in the cell nucleus of eukaryotic cells. 10. Question: what is the function of messenger RNA, mRNA? Suggested answer. mRNA carries genetic information from DNA to the ribosomes, where proteins are synthesized during translation. 11. Question. How does transfer RNA, tRNA, contribute to protein synthesis? Suggested answer. tRNA transports specific amino acids to the ribosome, facilitating their incorporation into the growing protein chain. 12. Question. What is ribosomal RNA, rRNA? Suggested answer. rRNA is an RNA molecule that serves as a structural component of ribosomes, where protein synthesis occurs. 13. Question. Define the term genetic code. Suggested answer. The genetic code is a set of rules that specify the relationship between nucleotide triplets, codons, in mRNA and amino acids in proteins. 14. Question. What is a codon? Suggested answer. A codon is a sequence of 3 nucleotides in mRNA that codes for a specific amino acid during protein synthesis. 15. Question. What are amino acids? Suggested answer. Amino acids are the building blocks of proteins, linked together in a specific sequence to form a polypeptide chain. 16. Question. Explain the term universal genetic code. Suggested answer. The universal genetic code refers to the fact that the same codons code for the same amino acids in nearly all living organisms, reflecting a shared genetic heritage. 17. Question. Where is DNA located in prokaryotic cells? Suggested answer. In prokaryotic cells, DNA is located in the nucleoid region, which lacks a membrane-bound nucleus. 18. Question. What is the role of guanine in DNA? Suggested answer. Guanine forms a base pair with cytosine in DNA, contributing to the stability of the double helix. 19. Question. How does the structure of RNA differ from DNA? Suggested answer. RNA is typically single-stranded and contains ribose sugar, while DNA is double-stranded and contains deoxyribose sugar. 20. Question. What is the significance of the helical structure of DNA? Suggested answer. The helical structure of DNA allows for the efficient storage and retrieval of genetic information and provides stability to the molecule. This content is mainly for the purpose of learning English vocabulary, English grammar, and English sentence structure. We are learning American English. A sincere thank you to everyone who watched. Thank you for considering subscribing to see more exciting videos.